In the past few lectures, from lectures 0 to 8, we took a look at how the human, how we can describe different biophysical aspects of the human body. So we took a look at different organ systems in our bodies as well as in plants, and tried to apply some physics to that. Starting from lecture 9 onwards, we're now going to try and apply physical concepts to smaller things. So instead of looking at tissues and organs, we're going to take a look at molecules and cells. So there is a completely separate area of biophysics that's called molecular and cellular biophysics. In this course, we're just going to go through some of the really generic topics that touch on molecular and cellular biophysics. If you're interested in learning more about each of these, then you can definitely take Physics 380, which only focuses on this subfield of biophysics. So first of all, we're going to go through a recap of the chapter about vision. That's the last part of human anatomy that we'll be talking about. I just want to present the most important equations that you should know moving forward, and then immediately pivot to an introduction to cellular and molecular biophysics. So that means understanding what that field is, as well as where it lies on the scale in terms of size. After that, we're just going to go through an overview of what types of cells there are, whether they're classified as prokaryotes or eukaryotes, animal or plant cells, and what kind of organelles there are in animal and plant cells. So you will have to know the biology for this, basically the structure and function of various organelles, but overall it would be fairly straightforward. After that, we take a look at the four macromolecules: proteins, lipids, nucleic acids or DNA, as well as carbohydrates. The course itself will focus specifically on proteins, DNA, and lipids as separate chapters, but we will be going through an overview of carbohydrates for this lecture. So let's go through the recap. In the recap, we're just going to go through some key equations for the biophysics of vision. There were a lot of equations presented in that chapter, but there's only so many you need to know. Afterwards, we're going to talk about very briefly the anatomy of the eye. And one of the things is we don't expect you to memorize every single part of the eye. We just want you to understand the importance of simplifying the eye so that we create the reduced eye model. So knowing where the reduced eye model comes from, as well as how to apply the equations to determine the accommodation of the eye, those are the key concepts to remember. Next, you have photoreceptors, so rods and cones. Knowing the difference between those are important as well. So some of the key equations, apart from the speed of light and photon energy, which are fairly generic, what I would want you to focus on would be the equations we used for the reduced eye model, or anything related to the reduced eye model. The reduced eye model comes from having multiple layers of refraction that take place. That means you have to understand what a refractive index is. You have to understand how much refraction occurs, which comes from Snell's law. And then, of course, if you're trying to focus on a specific object at a certain distance, the thin lens equation helps. And you can also determine the refractive power through this equation. So those are the key equations. In red, I have angular resolution here as a typical equation we would use to determine just the minimal distance that you can tell apart, but we will not be testing on that for this course. Okay, so for the reduced eye model, in terms of the anatomy of the eye, I just want to point out that in biophysics, we really want to try and simplify something so that we can create a model to mimic the real life situation. So when we take a look at the physical aspect of refraction in your eyes, we know that when light hits your eye, it first goes through the cornea in white, then it passes through the iris, then it goes through the lens, and then through the vitreous humor. So I can count how many layers there are. It passes through 1, 2, 3, and 4 different media. And so therefore, there's going to be approximately 4 calculations that you would have to do in order to calculate the total refractive power of all the lenses combined. So instead of calculating each of these separately every single time, we combine all of it and we use something called the reduced eye model. So that's when all of the refractive surfaces of the model of the eye are replaced by one representative lens. And that representative lens has a total refractive power of 60 diopters. So then what you can do is use that assumption or that model of 60 diopters to see how much the eye's overall power has to change to accommodate objects at different distances from your eye. So the eye will accommodate and change its refractive index to allow you to focus on objects at different distances. In terms of understanding how the accommodation works, just understand that your ciliary muscles are going to be either relaxed or contracted when you accommodate. So understanding what those ciliary muscles do would be important physiologically. Next, we took a look at the retina. So basically what happens is, going back to that slide with the anatomy of your eye, the light passes through and then hits your retina at the back of your eye. And that's where it converts the light that you see, or the object that you see, into an image that goes to your brain for processing. So the important part isn't really to understand the exact anatomy of this, is to understand that the retina contains special cells called photoreceptors. Rods govern your perception of black and white images, so it's going to contain something called rhodopsin. Because it perceives black and white, it means that it also is going to be more sensitive to light intensity. So the more bright something is, the whiter it'll appear. The darker something is, the more black it's going to appear. So rods play a huge role in night vision. In terms of cones, they perceive color and provide detail vision. So that would be important to note the difference between the two. The other thing I want to mention is that in terms of your photoreceptors, because they're so long and thin, they can be modeled as fiber optic cables where the light kind of goes in and bounces around as through total internal reflection. It's not something you have to know, but just something interesting. So first, we're just going to go through an introduction to cellular and molecular biophysics. So I want to just go through where this field is in terms of a size scale, and then go through why cells and organelles would be important. So over here, I have this diagram, and it goes from large to small. So what exactly is cellular and molecular biophysics? Before we answer that, we want to take a look at their relative size to each other. When we study biophysics, we have a choice. We can look at the macro scale, and even bigger than this would be tissue systems and the human body, which is what we've been doing in the past few lectures. So we took a look at different organ systems. These topics related to organ systems would be more beneficial to you if you're moving on to medical biophysics, because you're looking at the big picture. But there are also some with a more stronger research focus that want to study things on a small scale. So for those who are interested in smaller scale items, then you want to take a look not at the organ, but rather the tissues that make up the organ, potentially the cells that make up your tissue, and then the molecular structure of your cells and organelles. So once we cross into that realm of cellular and molecular biophysics, we then move on to just looking at simple molecules and atoms. So that's where those macromolecules would come from, and also different intermolecular interactions. So the way we're going to structure the next few lectures is really just, we're going to start by looking at intra and intermolecular forces. So that's going to be between atoms and basically how we would be able to create simple molecules and hold them together. Then through additional intermolecular interactions, you would end up being able to have those macromolecules to study. So your proteins, your nucleic acids, so on and so forth. Then after that, you can take a look at how they interact with each other, what kind of functions they have, and how different biomolecules interact with each other. So a lot of biophysicists um, are going to study biomolecular or molecular structures and then move on to cells. So there's an entire subfield of cellular and molecular biophysics that kind of focuses in on these three sections. So in terms of the examples and applications we can go through in terms of practice problems, there won't be a lot. The next few lectures really will just be focusing on the biological side of things. So understanding the different structures and functions and understanding why they come to be is more important than being able to model or solve an equation. Okay, so first an overview of the difference between the two types of biophysics, cellular and molecular. We're going to start off with the smaller version, the molecular biophysics. It's a subset of biophysics where biological problems presented are on the molecular scale. So we are just going to focus on interactions of biomolecules with other molecules, and of course how those biomolecules form as well. With cellular biophysics, we focus on the physical principles used in cell function. So that means we're going to determine which living system we look at first. We could be looking at plants, in which case the cellular makeup of the plant would be fairly different than the makeup of an animal organism. So there are two subfields within cellular biophysics, and there's more depending on what living organism you're studying. You can focus on plants, you can focus on humans, you can focus on animals. But the main thing is we first need to be able to differentiate between plants and animals. Even for us, we can just consider ourselves animals as well. So being able to differentiate between humans and plants on a cellular level is a first step to being able to understand the differences between the two on a molecular level. So an organism, the first thing we have to talk about is what is an organism? An organism is essentially any entity that exhibits the properties of life. So by this, we are simply just looking at something that's living. And living organisms can be broken up into two main categories. They can be prokaryotes or they can be eukaryotes. So there's these two. With prokaryotes, there used to be two kingdoms, bacteria and archaea. Now we just primarily say that bacteria are the main constituents of prokaryotes, while archaea is like a separate category. With eukaryotes, you have fungi, animals, and plants. So these are the three different types of living organisms under eukaryotes. But what are the differences between these two types? It's extremely important to understand that one of them actually has membrane-bound organelles, basically different compartments inside your cell, and the other type does not. So prokaryotes are the more simpler version of a life form. It means that they just have a cell membrane, and inside them, all of their components are just floating around in some sort of cytoplasm. So that means there's no membrane-bound nucleus, but rather just DNA, usually in a plasmid or something floating around. Then you have eukaryotes, and in that case, you do have membrane-bound organelles, and therefore a membrane-bound cell nucleus with a nuclear envelope. So just important to keep in mind that eukaryotes have membrane-bound organelles, while prokaryotes don't. So in our course, because we want to focus on biophysics, we want to focus on eukaryotes because they have plants and animals. The next thing, one of the main misconceptions, viruses. Where do they fall under prokaryotes and eukaryotes? Viruses are not living things. They do have DNA, but they essentially need to inject their DNA into a host, a living host, in order to reproduce. So an important thing to remember is that, with the coronavirus and everything, a living organism is defined as something that exhibits the properties of life. And one of the main properties of life is being able to reproduce without the help of external parties. So In terms of prokaryotes, bacteria, animals, and plants, we are capable of self-reproducing within our own bodies. So, humans can have babies, plants can germinate and everything. But viruses, they don't self-replicate. They actually have to inject their DNA into either a plant or an animal cell and then take over the genetic information delivery system, whether it's transcription or translation. They have to take over that in order to reproduce more copies of themselves. So they take over the host organism in order to self-reproduce, and therefore, by that definition, they are not living organisms. Okay, so now let's take a look at the different types of cells between prokaryotes and eukaryotes, as well as different types of organelles that show up in eukaryotes. And one of the most important things is that we'll be differentiating between prokaryotes and eukaryotes, but then when we talk about eukaryotes, you will have to know the differences between an animal cell and a plant cell and which organelles are unique to each. Okay, so a prokaryote, like we said before, it lacks a membrane-bound organelle. It's usually unicellular. Sometimes it's multicellular. But for example, this is going to be a typical bacteria with a flagellum and some hair or pilus on top, or pili. It's divided into two kingdoms, bacteria and archaea. And archaean cell walls do not have peptidoglycan, so that's the difference between bacteria and archaea if you're interested. We don't have to worry about it for this course, but primarily just know that bacteria will be a prokaryote. Now, for prokaryotic features, one of the things is that it has no nucleus and it has no organelles. So basically, it's just one membrane-bound unit with all of its components floating around in that cytoplasm. So usually its DNA is just seen floating around in here, called a nucleoid or a DNA. And that nucleoid is a nucleus-like region of the cell where your genetic material is kept. So even though there's no additional membrane that binds or compartmentalizes your DNA, it's usually found within a specific region in your cell. So that area is just called the nucleoid. So that's pretty much all you would have to know about a prokaryote, because our focus is going to be on eukaryotes. And eukaryotes are unique because they have a nucleus and organelles, and they're divided into three kingdoms, two of which we will be focusing on immensely, plants and animals. So what are some eukaryotic features? Of course, the main difference is we have organelles, so basically a bunch of different sections within your cell that are bound by a membrane. Then you have your nucleus as well, which holds your genetic information. So one of the unique things about eukaryotes as well is that they're usually multicellular with many cell types. We can reproduce asexually through mitosis, sexually through meiosis, and gamete fusion. So just as a quick overview, and you'll have to know this, but this is just a cross-section of a typical eukaryotic cell. All eukaryotes are going to have a plasma membrane. Between plants and animals, plants are going to have an additional cell wall, whereas animal membranes will not. You have lysosomes, secretory vesicles, and for a eukaryote, you have your nucleus, which contains a nuclear envelope with pores for transport, as well as ribosomes for protein processing. And of course, within your nucleus, you also store DNA. Outside of your nuclear envelope, you have your endoplasmic reticulum. There's both rough and smooth endoplasmic reticulum, and we're going to go through the function and structure of each. But they look fairly similar, and they're just many layers of membrane-bound sheets, essentially. Your rough endoplasmic reticulum has ribosomes on top attached to the surface, which is why it's called rough, whereas your smooth endoplasmic reticulum does not. So let's go through just the differences between animal and plant cells, and then we can go through some organelles. So I already named a couple of them. First of all, the previous picture didn't really show you what was inside that nucleus in detail, but here you have the nuclear envelope with your nuclear pores and your ribosomes on top. Over here, you have your nucleolus, which is the core of your nucleus, and around it you're going to have chromatin. So that chromatin is going to be your DNA or your genetic information. Apart from that, you have, for an animal cell structure, you have peroxisomes, you have lysosomes, which are unique. You have some other ones which we won't really worry about. You have mitochondria, which is also very important. They are the powerhouse of the cell, like I have on my shirt. And you have your Golgi apparatus or your Golgi complex over here, which I'll explain in a bit. So essentially the key things to your animal cell, you have lysosomes, which are unique, and then the rest are shared amongst plants as well. So looking at plants, you can see that the structure is very different. It's no longer irregular shaped, it's more rectangular and it's more rigid looking. So through a plant cell, you also have a nucleus. You have the same types of organelles until you go to here. So apart from that, you have the cell wall as well for the rigid structure and the plasma membrane. But what plants also have is the ability to go through photosynthesis. And because of that, they have chloroplasts. And so a chloroplast is going to have a thylakoid membrane, as well as different layers that are stacked on top of each other called the grana. They also have vacuoles. So vacuoles, it's important to know that there are vacuoles in both animal and plant cells, but in plant cells, there's a singular large central vacuole for energy storage. So we're just going to go through all of the key organelles now. So the key organelles in eukaryotes, the first one I want to talk about is the nucleus. So in terms of the nucleus, the structure is fairly straightforward. You have a nuclear envelope with nuclear pores. You also have a nucleoplasm in pink here that contains chromatin, and in yellow you have the nucleolus, which is the core. The function is to store genetic information as well as to assemble ribosomal units. I put the endoplasmic reticulum next because the endoplasmic reticulum is commonly associated with your nuclear envelope, and it's almost a continuation of it. So the endoplasmic reticulum is an extensive network of membrane-bound tubes and sacs. And it's continuous with your nuclear envelope. So usually this is just going to be attached to your nuclear envelope. There are two types of ER. There's smooth and rough. So your rough endoplasmic reticulum is going to have ribosomes attached to its surface. And then when there are ribosomes, usually what you can do is assume that they're going to have a role in protein synthesis. As a result, taking a look here, your rough ER is going to play a role in protein synthesis. But looking at the smooth endoplasmic reticulum, so over here, this picture actually represents rough ER because it's got the dots here to represent the ribosomes. In a smooth ER, you don't have any of this. You still have those membrane-bound sacs and tubes, but your smooth ER will not have them, and therefore it's going to rather participate in the synthesis of lipids because it contains various enzymes for lipid production. Next, we have the Golgi complex, basically very similar to the endoplasmic reticulum, but it's more compressed. You have stacks of flattened membrane sacs, which are called cisternae. The function isn't to create or synthesize proteins, it's to modify and process proteins. So that means creating your polysaccharides and glycoproteins, as well as managing their storage and release. Next, shared amongst all eukaryotes are mitochondria, singular mitochondrion. The structure is, it's actually got a double membrane, so you can't really see it here, but you have your first membrane, and then you have your second membrane. Your inner membrane is going to have infoldings called cristae, and then the function is ATP production. It stores a bunch of enzymes that catalyze redox reactions, and so ATP, or adenosine triphosphate, will be converted to adenosine diphosphate, or ADP. In other words, it loses a phosphate because a bond is broken, and that reaction is going to supply energy to your system. One thing I also wanted to mention about mitochondria is that they do have mitochondrial DNA and RNA that can participate in protein synthesis. Next, you have lysosomes and peroxisomes. Lysosomes are actually only seen in animal cells, and they're just a membranous sac of hydrolytic enzymes. So their purpose is to break down ingested substances, cell macromolecules and damaged organelles, and to recycle them. So in short, they help with digestion and recycling. You also have peroxisomes, which are present in both types of eukaryotes. They're very, very small. They also have enzymes, and they're usually used and involved in a variety of metabolic processes and reactions. So sometimes it serves to oxidate fatty acids, ethanol, or other compounds. So in terms of the importance of these two, I would say, just know about them. It's the other ones that are much more important. Now, the two over here are primarily found in plants. The first one is definitely only found in plants, whereas the second one, the vacuole, is found in both. So in terms of the structure of a chloroplast, it's a double membrane, so you can kind of see it here. Dark green, lighter green on the outside, that's your first membrane, the outer membrane. There's going to be some fluid, and that's called the stroma. And inside that fluid, you can see another membrane, thylakoids, stacked into grana. So this would be for plant cells only. So this would be a thylakoid. Each disc would be a thylakoid, and it's stacked up so that you have these different granum or grana of stacked discs. So the function of these would be very similar to mitochondria. It's but the reverse. It produces ATP and sugars via photosynthesis. With vacuoles, there's a large structure. It's a very large membrane-bound vesicle. In plants, you only have one large central vacuole, but in animal cells, you have many smaller ones. So they serve various purposes. They usually help with digestion, storage, waste disposal, water balance, cell growth, and protection. But in most cases, we just consider it a storage space. So hopefully through those few organelles and a few slides, you can kind of understand the main differences between plant and animal cells. But the last thing I want to touch on is the cell wall or the cell membrane. So animal cells are simply going to have a cell membrane, but no cell wall. Essentially, it's a plasma membrane. It will consist of transport and receptor proteins. And that cell membrane, very important to remember that cell membranes have selective permeability to allow you to maintain an intracellular environment. When we take a look at plant cells, though, they not just have a cell membrane, they also have an additional cell wall. And that cell wall is made of carbohydrate fibers that run through some sort of protein/carbohydrate matrix, or a glycoprotein matrix. So it offers protection and structural support. Other key differences I mentioned before, only plant cells have chloroplasts, and plant cells have one large singular vacuole, while animal cells have many smaller ones. Okay, so we want to start off with the four macromolecules now. And the four macromolecules are proteins, lipids, carbohydrates, and nucleic acids. Because we're starting our discussion of macromolecules, it's important to note that for this section, we're simply just introducing them. We won't be focusing on carbohydrates as much, which is why I'll focus more on carbohydrates for this lecture. But each of the other three macromolecules will have a dedicated lecture for it. And just keep in mind, if you are interested in modeling any of these macromolecules, Physics 380 is the course that will go through each one of these in a lot more detail for several weeks each. Okay, so the four great macromolecules. This slide is a slide that I present all the time when I talk about the different macromolecules. The four of them, just know their names and know their function. So with nucleic acids here, their main function is to store and transmit genetic information. Just think of DNA and genetic info to remember that. Then you have lipids. So this is a phospholipid with two tails. But in general, lipids are going to store long-term energy. It protects via formation of barriers and membranes, so it does play a role in protection. Next, you have protein. So over here, I have a huge globular protein. It doesn't have to be a globular protein, but proteins do most of the work in the cells, and they're going to help with the structure, the function, and the regulation of your host body. So there are many, many proteins. They can interact with different macromolecules, and those interactions will give rise to specific functions. So the last one we want to talk about is carbohydrates. So carbohydrates are used for energy storage and, most importantly, surface properties. So when we talk about proteins, when we talk about lipids, and when we talk about nucleic acids, usually there are carbohydrates involved through surface interactions. So with lipid membranes, for example, carbohydrates can be attached to lipid membranes on the surface, which may act as a specific marker for other enzymes or proteins to attach. So carbohydrates, in terms of their role, is usually combined with different functions of specific macromolecules. So let's first start with an overview of carbohydrates. So first of all, carbohydrates should be a very familiar term because it's used everywhere in health and nutrition, and we hear it all the time. So usually when we eat, we always hear whether somebody's going to go for a high carb diet or a low carb diet. It's usually associated with our conception of weight loss, weight gain, and diet. And in general, the idea is fairly similar from a biophysical standpoint or a physiological standpoint. What carbohydrates do is they provide energy to your body, specifically through glucose. And glucose is an ingredient in many staple foods. And so any starchy foods are going to have a lot of glucose or sugars. And when you break down sugars, it's going to give you energy. So the problem is, where is this energy stored? It's actually in the chemical bonds between its carbon atoms. So the first stage in the breakdown of glucose is called glycolysis. It's not something you will have to know. Glycolysis, or the splitting of glucose, occurs in many steps. There's basically 10 steps that are fueled by enzymes. The second stage occurs in your mitochondria. So one carbon atom and two oxygen atoms are going to be removed, yielding more energy to your body or your system. And so essentially a few more processes are going to convert that energy into a usable format for your body. So apart from providing energy to the body through glucose and glycolysis, carbohydrates also provide other important functions in plants, animals, and humans. So I'm just going to talk about these three briefly, and I would say these are important to know in terms of what functions carbohydrates have. So one of the things that carbohydrates can do is store energy. If you have too much glucose, then usually it's stored as glycogen, which is basically a chain of many sugars attached to each other. It's a polysaccharide. So the majority of glycogen is actually stored in your muscles and in your liver. And one molecule of glycogen might have over 50,000 individual glucose units that are ready to use whenever you want. So that in terms of glycogen, it also, well, there's going to be a picture afterwards, but you'll see that glycogen is a very branched polysaccharide. So because of all the branches, you can easily process the glucose units inside glycogen by just picking away at the different branches versus a long string. Next, so that's energy storage. Excess glucose is stored as glycogen. Next, we can talk about the building of macromolecules. So even though we have carbohydrates, and most of the absorbed glucose is used to make energy, some glucose is going to be converted to ribose and deoxyribose. And those are essential building blocks of important macromolecules like RNA and DNA, respectively. So glucose is also used in the it's used for protection against oxidative stress because it also makes the molecule NADPH, as well as other chemicals in the body. So that's another important aspect of carbohydrates. They're not just meant for energy, and they're not just meant for energy storage. They can be used to fuel other processes within the body. And of course, they can be converted themselves to be used in the construction of macromolecules like DNA. Next, you have lipid metabolism. So that really has to do with blood glucose levels and insulin. And so I'll touch a little bit on diabetes here. As your blood glucose levels rise, what happens is the use of lipids as an energy source is inhibited. So basically, too much glucose means that you're going to use the glucose versus your lipids as your energy source. The reason for that is because if you have too much glucose in your blood or too much sugar in your blood, it's going to stimulate the release of a hormone called insulin, which tells your cells to start using glucose instead of your lipids to make energy. So if you are unable to regulate your insulin production and release, then you end up with issues related to diabetes. So then we've talked about what carbohydrates are and what they can do, why they're important. Now we should probably take a look at what they look like. So over here, I actually have the basic building blocks of all carbohydrates, monosaccharides. So with monosaccharides, you can have glucose, fructose, galactose, deoxyribose, and ribose. So they're just some examples. The general formula for monosaccharides is going to be CN, H2N, ON, and that's in general. There are some exceptions. The number of carbon atoms helps derive the suffix of the name. So for example, if you have 3 carbon atoms, then it's going to be some sort of triose. If you have 5 carbon atoms, it'll be a pentose sugar. Next, you can also see what kind of functional group is attached to your monosaccharide. If you have an aldehyde functional group, then you end up with an aldose. And if you have a ketone functional group, then you'll end up with a ketose sugar. So in general, just understand that monosaccharides are the building blocks of carbohydrates, and that they can have 2 different types of functional groups for classification. Now, what happens when they come together? As a building block, they have to create longer chains. So a monosaccharide will eventually become a disaccharide and eventually a polysaccharide. So a disaccharide, which is also called a double sugar or a BIOS, is the sugar that's formed when you have two monosaccharides that are joined by a glycosidic linkage here. And the reason why that glycosidic linkage is formed is because of a dehydration synthesis. So your OH, your two hydroxyl groups are going to combine. H2O is going to be kicked out, and you're left with an O that connects the two together. So that is your glycosidic linkage. And another important aspect to remember is that even though they are now two monosaccharides together as one, they are still simple sugars, like sucrose and lactose. They're still simple sugars that are soluble in water. So the last part is if you have a bunch of monosaccharides that are connected to each other, you end up with a long chain or branches of polysaccharides. Some examples are starch and glycogen. So glycogen is actually presented here. And remember I said glycogen is very branched so that it provides easy access and additional surface area to allow glucose subunits to be broken off and used more efficiently. So there might be an important question about why glycogen is structured the way it is. Next, we want to talk about where macromolecules exist. So we did take a look at carbohydrates. We took a look at the different types of macromolecules, proteins, DNA, and lipids, and they all exist within your body. So if they exist, they have to be in some sort of solvent, and we assume that solvent to just be water. And we make that assumption because we want to make our lives easier, especially when we're trying to model something. So we know that the amount of water in a cell is about 65%. The amount of water in blood is 80%, like from our previous studies of the cardiovascular system. The amount of water in fat cells is a little bit less, 10 to 20%. But overall, there is a lot of water in a living organism. And because of that, your macromolecules must exist in a water-like solvent. So we can assume that when we're modeling all of these different macromolecules, they exist in bulk water. And we just have to realize that water is a polar molecule with a dielectric constant of 80. And because of hydrogen bonding and its electronegativity, it allows for a good environment for different substances to dissolve. So we'll be talking about polar molecules and hydrogen bonding in more detail in the next lecture. But for now, we should understand why water is important to our bodies. So because of water and its electrostatic characteristics, number one, it helps to dissolve and stabilize biological molecules and ions. So that makes sense because ions are going to have charge to them. And because water also is polar, therefore, both of them will want to mix together. So for example, polar substances are going to dissolve in polar solvents. Now, it also controls temperature balance in organisms by absorbing and releasing heat. It adjusts in intracellular pressure to help maintain cell structure, so that would be osmosis and other pressure gradient related mechanisms. It also helps form structural layers between polar ends of proteins and lipids. So for example, membranes, because of its polar properties and because lipids are amphiphilic, they have a polar and a non-polar section. They help to organize the lipids and self-assemble them so that a biomembrane is formed. Water is also important. Apart from animals, you have plants. And remember, we had that chapter with plants where water played a key role in transporting nutrients up and down the roots and throughout the plant. So that's important to know as well. Because our blood has a high concentration of water, it also means that water plays a crucial role in the cardiovascular system and therefore blood circulation. After that, you also have the fact that water participates in many different reactions, whether it's photosynthesis, cellular respiration, where it's oxidizing and donating electrons. So in terms of understanding this, just be able to pick out which different functions belong to water and which ones do not, if there's a mix and match question of some sort. Okay, so just as a summary, what should you remember? So I want you to remember the difference between cellular and molecular biophysics and why it's important, where it falls on that scale of large to small. After that, just understand the different cell types, understand the difference between a prokaryote and a eukaryote, understand the difference between plants and animal cells, primarily the cell wall in plant cells. And then, of course, be able to determine which organelles belong to which type of cell, plant or animal, if some organelles are shared amongst the two, or if some organelles are unique to specific types of cells. Knowing the structure and function of those organelles would also be important. Afterwards, we talked a lot about the four macromolecules, specifically carbohydrates. Just be able to name and list the function of each macromolecule, be able to understand the importance of carbohydrates, their function, and their structure from monosaccharides to disaccharides to polysaccharides. And of course, being able to understand the importance of water and why we choose water to be a good model for as a solvent for all macromolecules. Apart from that, there are no problems or mathematical problems for this chapter because it's primarily based in biological concepts and physiological concepts.